Hi everyone, let's get started. Today we're going to be talking about microbial genetics and the processes of transcription and translation. From last time, you remember, when we were discussing DNA replication, we said that DNA replication makes new DNA from old DNA. We described this as being a semi-conservative process, where each strand of the old DNA served as a template, and new DNA is built right on top of that template, adding bases to the new strand so that they are complementary to the bases in the old strand. We also talked about how we are able to perform our own version of DNA replication in the laboratory, and we used the phrase in vitro to describe that. Remember, when we say something is in vitro, we're saying that it is outside of the living organism. In other words, we're doing it in the laboratory and not inside a living cell. Polymerase chain reaction, or PCR, is the in vitro form of DNA replication. Now, we have to remember that DNA replication is not the only thing that living cells do with their DNA, and microbes are no exception. Microbial cells and other cells also use their DNA for gene expression. We have to go back to the central dogma to remind ourselves of these processes. DNA is used to make RNA through the process called transcription, and RNA is used to make protein through the process of translation. And recall, it's the proteins that give microbes and other organisms their traits. Now you'll notice I've added in a little bit of extra information in the first statement here, that DNA is used to make RNA through transcription. We need to remember that there are three different kinds of RNA. There's what we call mRNA, or messenger RNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA. All three types of RNA are made through the process of transcription, but only mRNA is translated to make a protein. So, messenger RNA we often refer to as the transcript of the gene. Think about the example of a courtroom proceeding. There's always a person making a transcript of what happens in the courtroom. The judge and the defendant and the prosecutors, they're all doing the case in real time. They're all speaking out loud and discussing the case in the courtroom. But then there's a court reporter who is making a transcript of the case, a person who is recording the spoken language down in a written form. The same thing is happening during transcription in a cell. The DNA is the sort of original discussion, the original format, and the messenger RNA is the transcript. It's essentially a copy of the DNA. We know it's not an exact copy, it's a complementary copy, but the idea is the same. It will be used to carry the message or the record of the DNA to the ribosome, so the ribosome can translate that message. rRNA, if you recall, is ribosomal RNA and is part of the ribosome, both the small subunit and the large subunit. And rRNA is going to assist the ribosome in performing its function, which is translation. tRNA, or transfer RNA, is also involved in translation, but tRNA is going to behave like a middleman. tRNA is going to connect the information in the mRNA to the amino acid. Going back to our courtroom example, when you make a transcript of a conversation, you make a written recording of it. If you want to translate that recording, you take it in one language and you turn it into another language. That's what translation is all about. So in the case of genes, we're taking the transcript, the mRNA, which is written in nucleic acid, and we are translating it into the language of protein. And the tRNA is going to be an assistant in that process. The tRNA is going to help match the codons in the mRNA to the amino acids. Now I want to take a couple minutes and talk about the structure of RNA. Just like DNA, RNA stands for something. It stands for ribonucleic acid. And similar to DNA, there's a subunit in RNA, something that repeats over and over again. And there's a drawing of this in the lower right-hand side of the slide. And right away you can see that this subunit, this nucleotide, looks a lot like the nucleotide that we saw in the DNA molecule. There's a sugar in the middle, there's a phosphate group, and there's a base. Now, one of the first things that is different between the subunit of DNA and the subunit of RNA is the base. Remember, the base in DNA might be a guanine or a cytosine or an adenine or a thymine, but in RNA there is no thymine. Instead, there's a very similar looking base called uracil. So instead of having A's and T's and G's and C's, in RNA there's A's and U's and G's and C's. That's the first difference. The second difference is in the sugar. Remember that in DNA, we refer to it as being deoxy ribo or ribose. This is plain old ribose. So this sugar looks a little bit different from the sugar that we see in DNA. In DNA, there is a carbon number 2 here that has no hydroxy group on it, and carbon number 3 here that has a hydroxy group on it, or a hydroxyl group on it. In RNA, both the carbon number 2 and the carbon number 3 have hydroxyl groups coming off. So there's a structural difference in the sugar between DNA and RNA. It's called deoxy ribose in DNA, and it's called ribose in RNA. Now there's one more structural difference that we need to be familiar with between the DNA molecule and the RNA molecule inside microbial cells, and that's this. DNA exists as a double-stranded molecule in a living cell. RNA doesn't. RNA exists as a single-stranded molecule. DNA, remember we said, gets into a very complex folded up structure called a nucleoid. RNA also folds, but it folds with itself. And we'll take a look at that in just a minute. So on this slide, we're reminded about the structural differences between DNA and RNA. Up at the top, you can see 2 sugars. So this is a carbon number 1, carbon number 2, carbon number 3, carbon number 4, and then off of the ring is carbon number 5. This is the sugar that we saw in DNA, and we called it deoxy ribose. The ribose sugar has 5 carbons in it, and this is called deoxy ribose because it's missing an oxygen. Normally we would see an OH on carbon number 2 and an OH on carbon number 3, but we're missing an oxygen, so it's called deoxy, or without oxygen. Over here on the right hand side you can see ribose. Again, a 5 carbon sugar, and this time we've got a hydroxyl group on both carbon 2 and carbon 3. So this one is just plain old ribose. This sugar is found in DNA. This sugar is found in RNA. Down here, we're taking a look at a couple of the bases that are found in DNA and RNA. The one on the left is thymine. The one on the right is uracil. Now, we don't have to know the structural features of the bases in DNA and RNA. We just need to understand what bases are present and which bases complementary bind with each other. Now, it is interesting to note that when you compare thymine and uracil side by side, they almost look identical. The difference is way up here on this carbon, you can see that there's a methyl group attached in the thymine base, and over here there's just a plain old singlet hydrogen attached. So there is a difference structurally between these two bases, but boy, they sure look similar. We find thymine in DNA and uracil in RNA. So both thymine and uracil would bind to A, or adenine. And here is the reminder about the single-stranded nature of RNA. DNA exists as a double-stranded molecule. In its double-stranded form, in its double helix form, DNA is very stable. RNA is different. RNA exists as a single-stranded molecule. It will fold onto itself wherever it can complementary bind to itself, but overall it maintains itself in the cell as a single-stranded molecule. DNA, if you think about it, has to be structurally sound. It has to be stable because it contains the genetic information for the cell. It's arguably the most important molecule in the cell. RNA is different. RNA doesn't have to last forever. RNA is just necessary for that particular time when a protein needs to be made. And so RNA doesn't need to live forever. It doesn't need to be a stable molecule, and it isn't. All right, so during the time of gene expression, when a microbial cell has a need for a particular protein, something has to happen to that DNA molecule, that nucleoid. The very first thing that has to happen is the area in the DNA where the gene is has to be accessed in order to be transcribed and translated. So DNA has to be unwound in that area, wherever the gene is. Now this work is done with an enzyme, and you're going to see that a lot of different enzymes are involved in transcription and translation, each enzyme having its own particular job to do. We don't need to know all the names of all of these enzymes. We just need to understand that these processes require an enzyme to happen. So, in the case of transcription, the process of building the RNA from the DNA is performed by an enzyme called RNA polymerase. You saw in DNA replication that the enzyme was called DNA polymerase. So these are both polymerase enzymes. They are going to make polymers. They're going to take subunit molecules called nucleotides, and they're going to attach them one to the next, to the next, to the next, to the next. Each one of those nucleotides you can think of as a monomer. And when you build a long chain of them, you've created a polymer. Poly, remember, just means many. So DNA polymerase is the enzyme that makes the polymer of DNA. And RNA polymerase makes the polymer of RNA. Now, recall our definition for what a gene is. A gene is a piece of DNA that contains two different kinds of code. There's code in there for building a protein, and there's what we call regulatory code. Code that is there to determine if a protein is going to be made, when a protein is going to be made, and how much of that protein is going to be made. we're going to need to start learning about the regulatory regions of genes when we talk about transcription. In order for transcription to begin, that important enzyme, the RNA polymerase enzyme, has to bind to part of the regulatory region. And this is a portion called the promoter. The enzyme is going to be guided in and it will bind with this promoter region so that it is positioned right at the start of the coding region for the protein. As the enzyme is brought in and it binds to the promoter, this is what we refer to as the initiation step of transcription. The enzyme will then move forward through the protein coding region of the gene, adding nucleotides one at a time in a complementary pattern according to the old DNA. In other words, where there's an A in the DNA, the enzyme will place a U in the RNA. Remember, it would normally have put a T, right? But there's no T in RNA. So wherever a T would be located, a U is substituted. If there's a G in the DNA, the enzyme will place a C in the RNA, and so on and so on and so on, all along the length of the protein coding region of the gene. This process is called elongation. Now, something else to note here. Just like we saw with DNA replication, during transcription, RNA can only be made in one direction. It can only be built from the 5' to the 3'. And remember what that means to us. That means that bases can only be added, ribonucleotides, in other words, can only be added to the 3' end of that growing chain of RNA. Unlike DNA replication, however, transcription is only going to head in one direction along the length of the gene. Remember, during DNA replication, once that replication bubble opens, replication occurs in both directions, all the way around the circular chromosome. But in transcription, we don't need it to go in both directions. We just need to transcribe that gene, that one gene that codes for that one protein that the cell needs at this time. Transcription will stop when the RNA polymerase enzyme reaches the end of the gene. And you might wonder, how does it know that it's at the end of the gene? Well, there's going to be a region in the DNA called the termination signal. And as the RNA polymerase transcribes the termination signal, that will end transcription. That's a good place for us to stop this video. We'll see you in part 2.